Good day, dear colleagues. I'm Klachkov Vladlen from Volgograd State Medical University, and my work is novel Quinazoline NCA1 inhibitor VMA2110 inhibits interleukin-6 secretion during LPS-induced inflammation. NHA1 is a validated drug target for cardiovascular and ocular diseases with cytoprotective, anti-ischemic, and anti-inflammatory properties. Due to its role in immune cells, we focus on a search of novel NHA1 inhibitors with anti-inflammatory properties. In our previous studies, we identified compound VMA2110 with potent NHA1 inhibitor activity, so we further investigate its inflammatory properties in vitro and in vivo. On the first stage, we isolated primary murine macrophages from peritoneal cavity, incubate them for 24 hours in plates, and then stimulate with LPS. After 24 hours, we determine cell viability, interleukin-6 and interleukin concentration. Acute lung injury during bacterial and viral infections remains a major healthcare problem, which was further highlighted by the recent COVID-19 pandemic. So on the second stage, we used this method to challenge the investigated compound. We found that VMA2110 inhibits LPS-induced nitric oxide synthesis and interleukin secretion with IC50 of 72 and 51 micromoles respectively, with no significant cytotoxicity up to 100 micromoles. In early model, dexamethasone and VMA2110 normalized amount of leukocytes in bowel, lung permeability index, and decreased neutrophils count and lavage. Biological examination of the lung 24 hours after lipopolysaccharide administration revealed massive infiltration with polymorphonuclear neutrophils in the intestinal tissue and purulent exudate in the lumen of the alveoli and pulmonary bronchioles. Immune cell infiltration was associated with diffuse alveolar damage and intestinal edema by congestive atelectasis. After administration of dexamethasone to LPS-treated animals, moderate thickening of the intra-alveolar septa was observed due to edema and accumulation of macrophage and macrophage-like cells in the intestinal tissue. Lung tissue of animals administered with VMA2110 was characterized by limited focal infiltration of the intra-alveolar septa with neutrophils. We have shown that NHA1 inhibitor VMA2110 prevents pro-inflammatory cytokine overproduction in cellular and animal models. These findings may guide the further development of protective agents for severe infections, including COVID-19. Thank you for your attention.